Hiya. Okay, so this is section 2 of DNA and proteins, and we're going to be covering protein synthesis.
Okay, so in this video we're going to learn about the different types of proteins made by cells and their various uses, and how proteins are made in a cell using DNA and cell organelles. So by the end of this you should be able to name the 5 types of protein in the body and describe their uses, state where mRNA is made, state where mRNA travels to, name the organelle where protein synthesis occurs, and name the subunit that makes up protein.
Okay, so proteins will start off with why they're important. They are essential molecules. Now, you might have heard things like fingernails are made of protein, hair are made of protein. Don't refer to those at National 5 level, okay? We're talking about cellular proteins. So there are loads of types of protein, including enzymes, hormones, receptors, antibodies, and structural proteins. Now, these proteins are keeping you alive right now. You take away any one of them and you're probably going to either collapse into a pile of mush, die instantly from hydrogen peroxide overdose, suddenly miscommunicate a signal in your body. So they are keeping you alive right now. They are absolutely essential. The first 4, so enzymes to antibodies, we will look at in much more detail later on in the course. Now, it might be later in this unit or during the multicellular unit, so they are very, very important. Structural proteins, the last one, we only really mention here. We don't really talk about anywhere else in the entire course. Okay, so we're going to have a quick overview of these 5 protein types, and then we're going to look at how they're made.
Okay, so firstly, hormones. It's probably something you've heard of before. It's something that most of you will be very aware of. They are types of proteins, unsurprisingly. So they're actually known as protein chemical messengers, and their job is basically to signal cells to do specific things. So one that you probably most commonly know is insulin. Insulin is a hormone that you have, and its idea is to tell your body all about regulating blood glucose, which we will touch on a lot in the multicellular unit. But the idea is hormones have a really specific shape, and that is so as they fit into receptors on a cell, a bit like jigsaw pieces. You can see in this diagram here with the hormone fitting the shape of the target cell. They have roles in lots of things in the body, including reproduction, which again is a key topic we'll talk about in the multicellular unit. But for now, hormones, good example of proteins that are just chemical messengers.
Okay, receptors. So we looked at the hormones. The receptors are also made of protein, and they live on the cell membrane, and they'll bind to the hormones to activate their signal. And they have got a specific shape in order to fit specific hormones. Different shape of receptor will work on a different hormone. So it's not like there's a one-size-fits-all theme. Now, there should be a theme that you see arising here with the proteins, and that's to do with shape. Okay,
so third one, antibodies. Antibodies are proteins made by immune cells to disable infections in the body. So if you are ill, these are the kind of things that are going to be helping you get over that illness, essentially. And again, it is all to do with a specific shape. So each antibody has a specific shape depending on what infection that you have. But again, talk on this much more in the multicellular unit.
Structural proteins, so this is the only time we're really going to talk about them. They help keep tissues in the correct places. You've got a muscle like a bicep on your arm. You don't want it falling down into your wrist or into your pinky. That'd be ridiculous. So quite a lot of structural proteins help keep tissues where they're supposed to be. There's one particular one, collagen in your skin is long and elastic, and that helps to keep skin tight and avoid sagging or wrinkling. UV damage damages collagen, which is why people who go out in the sun a lot will look older faster, because they damage the collagen in their skin, they get more wrinkles. Older people, the collagen is looser, which is why you can see Jeremy Clarkson's face flapping away there, is because his collagen is a lot looser, stopping keeping his skin so tight in the face of the wind, and as a result his skin's getting a bit saggy and a bit flappy. Now, you don't need to know an example of a structural protein, you just need to know structural proteins keep tissues in the correct places. Okay, but that's the only time we're going to talk about structural proteins, so it's worth remembering. Those are the 5 types of proteins. Structural is one of them.
Okay, so the final one, this is a really, really big one, a really important one, and the one we're going to talk about in the next key area, and that is enzymes. So enzymes are a type of protein that speeds up different chemical reactions in organisms, including humans. Without these, we will not survive. Without enzymes, the things that happen in our body to keep us alive will not happen quick enough. So enzymes, so crucial to survival. So really important type of protein. Again, they are specifically shaped to carry out a specific reaction, which we will touch on much more when we look at at enzymes in the next key area.
Okay, so those are the 5 types of proteins. Let's look at how those proteins are actually made. Now, a thought to keep in your head during this is the idea we just looked at those 5 types of proteins. They work well because they are the correct shape, because they probably have the right order of amino acids. So we're going to focus on that. So all of these proteins are produced in the ribosome by cells. Remember the ribosome, that little full stop type thing that's attached onto this big green block here, that is a site of protein synthesis. And the proteins are made using the code from DNA in the nucleus. So the nucleus has the code, the ribosome has the building site.
Okay, so protein synthesis, just to make sure you're all aware that to synthesize something basically means to build or create it. So this is all about just simply building, making proteins. And it's all to do with the process in which the DNA code is read. Because remember, the DNA code, that is the thing that actually carries the instructions for our proteins. It is those bases, that adenine, thymine, cytosine, and guanine, that codes for the proteins. And this whole process is about taking that DNA, reading it, taking it to a ribosome, making the correct protein. And very crucial, the order of the bases is everything. Different order of bases, different protein is made.
Sorry, I didn't know that was going to come up.
It's fine.
Okay, so the order of the DNA bases codes for the production of a specific protein. Different order of bases, different protein. Every group of 3 bases codes for an amino acid. That's a higher idea, so you don't need to worry about that too much. But when the amino acids join together, they become a protein. So if you look at this picture down at the bottom, is all those individual units. Those are all amino acids chained together, make a protein.
Okay, so recap on stuff again. You should already know, where is DNA found? It is found in the nucleus of a cell. In all the cells that have nuclei, that is where the DNA is. Proteins, they're not produced in the nucleus. They are produced in the ribosome. Those little dots just floating about the place. So really, really important you remember this fact. DNA in the nucleus, proteins are produced in the ribosomes. 2 very different places. Bit of an issue here. But your body has a way to get around that. So really important that you know this DNA never ever leaves the nucleus. DNA physically cannot leave the nucleus. It's not allowed to. It's a big no-no, because as soon as it's not in your nucleus, that cell has not got the right DNA to control all activities in its nucleus. So that cell is just as good as gone and dead. So DNA can never ever leave the nucleus, but it has to be in the ribosome at the same time. So something has to manage to copy that code for it. And there is that something.
And that thing is called mRNA. Now the little m stands for messenger. It's a good idea to try and remember that the m needs to be a little m. I don't know if they penalize in exams or not. You don't need to ever call it messenger RNA. mRNA is fine. Okay. Now the idea is mRNA is quite similar in structure to DNA, except it's single-stranded, and what it's going to do is it's going to carry a complementary copy of the DNA code. This means it is going to copy a matching copy of the bases of the DNA code. It's not going to be identical. It's not a photocopy. It's a complementary copy, which is why those words are underlined. If you don't quite understand the process after this, just try and memorize that sentence. mRNA carries complementary copy of DNA code. Now, mRNA can then take this code from the nucleus to the ribosome, and then we don't have to worry about the DNA leaving the nucleus and then the cell entirely collapsing. So think of it like, I don't know, the instructions for building a house are in one office, but the building site for the house is obviously away in the countryside. So the good idea would be to take a copy of the instructions for the building the house from there to the actual building site, okay? But you don't take away the original copy, because then nobody else can access it.
Okay, so in protein synthesis, there are kind of 4 basic steps to making a protein that, at this level. So the first thing is that the DNA will unwind, so that double helix will unwind and become almost like a ladder. And then actually at the gene that it wants, that will separate. So it will go from being a twisted shape to being a straight shape to then opening. So you have basically 2 free running strands of DNA where the gene that we want to copy is. The mRNA will then create a complementary copy. So basically it'll go along and go, okay, there's an A, there's a T, there's a C, there's an A, there's a T, there's a C, and match it up as it is. So it will make a complementary copy, so a matching copy. The mRNA, once it's got that full copy, it will then travel from the nucleus to the ribosome. So the DNA is never leaving, it's the mRNA that is leaving the nucleus. And once it's at the ribosome will read that mRNA, and then it will call in for the amino acids that it codes for and makes a protein.
Okay, so let's have a look at that in pictures. So the gene to be copied exposes bases. So what we have here, we have the chromosome, the DNA. The DNA has unwinded at a specific gene, so not the entire chromosome is unwinded, and it's exposed all of these DNA base codes. Okay.
The mRNA will then create a complementary, so a matching copy of the DNA. So you can see that's appeared here. It's got all the matching bases, except this is wrong because it's got U's instead of T's.
That's a higher idea. Don't worry about it. It's just looking for a complementary copy.
Yeah, so where it says U, imagine it says T for thymine.
Okay, and then in steps 3 and 4, the mRNA is going to go to the ribosome, and then the mRNA code is used to assemble the correct amino acids into a protein. So the idea is the ribosome will read the mRNA code and then build the right amino acids in the right order.
Okay, so summary of protein synthesis. So the end of key area 3 in unit 1. So 5 different protein types: hormones, enzymes, receptors, antibodies, and structural. If you do struggle to remember them, SHARE is a nice acronym for them.
Oh.
So SHARE, structural, hormone, antibody, receptor,
nice.
Enzymes. One of my 4th years taught me that.
Ah. So gene, we've said this before, but it's a section of chromosome that codes for a protein. It's mentioned here again because it's important.
mRNA, complementary copy of the DNA that travels from the nucleus to the ribosome.
Those 2 locations are quite often asked about, so it's good to have that memorized. The ribosome, as we know from key area 1, is the site of protein synthesis.
And amino acids, they are those molecules that when joined together in a specific order create a specific protein.
Okay, like a hormone or an enzyme or receptor, etc. So, things you should be able to do is your 5 types of protein. Remember, SHARE.
Structural, hormonal, antibody, receptors, and enzymes.
Very nice. Where is mRNA made?
It is made in the nucleus.
Where does mRNA travel to?
It travels to the ribosome.
What is the organelle where protein synthesis occurs?
The ribosome.
What's the subunit that makes up protein?
An amino acid.
Very nice. See? Easy. So that's the final bit of key area 3, DNA and proteins. The next key area is called proteins, but really it's all about enzymes. So we will see you there.